Whether it's your first mini prep or what feels like your 4 millionth mini prep, it's always to your benefit to know what's going on behind the scenes in every step. And you can actually do it yourself and make the components yourself, but it's a lot easier. Often we just go directly to a kit. And so these kits come from different vendors, and what they all have in common is that they're going to allow you to purify a plasmid out of bacterial cells. So you have some culture where you have bacteria that contain the plasmid that you're interested in purifying. They also contain a bunch of other stuff, including their own genome. The goal of the mini prep kit is to isolate just that plasmid, purify everything else out, even that bacterial DNA. And it's a really cool way in which it does this. And ultimately, it's going to use some sort of like column like this. And so these are typically done in, you spin them in a microcentrifuge. You can also stick them on a vacuum manifold, at least for some of the steps. And so this is the column. You want to avoid touching the bottom of the column. Here is a membrane. That's what's actually going to bind to the plasmid and let other stuff through. And then we'll make our plasmid soluble and allow it to go through. But it's going to come out of this little spout. And so you want to make sure you're not, when you take this out of here to like dump out your waste, so this is for the waste, when you're dumping out your waste, you want to make sure you're not actually touching this. And so you want to pull straight out of here without doing it. So you might need to do it carefully. I like to have some sort of waste beaker where you can just pour your waste into that and take care of it properly. And so then you have your column, and you are going to basically wash it with different solutions, and you're going to be pulling the liquid through either during centrifugation or during the vacuum filtration, and then that'll be dumping out of that. And then when you're ready, you're going to take it to a tube that you've nicely labeled with all the details about the plasmid, and later you'll add the concentration and stuff, and then you're going to spin it and elute it, so get it off into that nicely labeled tube that you have. So I'm going to take you through some of the steps of the mini prep kit, and this again, like I'll share with you the link where you can actually go and find what's in each of the different buffers and make it yourself, as well as you can order these tubes in bulk, and so make it really cheap. And so I'm not trying to like be a commercial vendor for anybody or anything like that. I just want to tell you what's going on in the steps. And this is really important because not only does it make it more fun to think about what the molecules are doing while you're doing your mini prep, but it also is going to help you understand how to troubleshoot where things might go wrong, and why we're doing things the way that we are, and how you can kind of cheat the system and do it yourself. So let's go through. So you're going to see when you open the box, you'll see a lot of different components, no matter what box you open. And so there's going to be a variety of different buffers. And I'm just going to take you really quickly through what each of these do, and then I'll take you in more detail. So the first thing is that you need to isolate the cells. So right now you have some sort of starter growth. And so typically you go from a plate where you have a colony on a plate, and then you want to make even more copies of the plasmid, because our whole purpose here is to isolate that plasmid. We want to make a lot of copies of that plasmid, and so typically we have these in bacteria that are good for making lots of copies of plasmids and are good for keeping it stable. So these are going to be cells like your XL1 cells or your DH5 alpha cells, not the expression cells, but the cells that actually have really high fidelity copying or polymerization. What you want to do now though is isolate them. So you took the colony, then you grew up even more of the bacteria in an overnight culture, typically like 5 mils in a tube. You can just take a pipette tip and swap the colony, drop it in your tube of the media. And so the pipette tip is actually like still even in here. So you want to make sure you're using sterile pipettes, and that the media has the antibiotic that you're using to select for them. And so you want to have your negative controls too, to make sure that your antibiotics are working and your bacterial media is not contaminated, so that only the bacteria that actually have the plasmid that has the corresponding antibiotic resistance gene will be able to grow. So you have these cells, and now you're going to spin them down. So for this, you're going to use the bigger centrifuge, not this little tiny one, because you need to spin down the tubes. So you spin them down, typically like 10 minutes. I just do like 10 minutes max speed. Doesn't matter too much. You just want to pellet the cells and get rid of the media. You want to get rid of the media as soon as you can after you pellet the cells. That's going to be a recurring thread. Whenever you centrifuge something, you want to get rid of the media. If your goal is to isolate the pellet, or even if it's not, you want to separate the pellet from the supernatant, the liquid above the pellet, as soon as possible, so that the pellet doesn't go into the supernatant, doesn't dissolve back. So once you have that pellet, now it's time to resuspend it and transfer it to a microcentrifuge tube. For every culture that you have, you're going to need a few things. One is going to be the column that comes with the kit. You need a final tube that's going to be nicely labeled. But you're also going to need a temporary tube, and that temporary tube is going to be for the point at which after you break open the cells, you're going to spin them down again. But this time you're going to spin them down in a tiny tube. So you're going to break open the cells, and you're going to remove the media, and that's just waste media, so you just want to bleach that. Then you're going to take that pellet, and you can actually resuspend the pellet and transfer it to a centrifuge tube, and then you're going to lyse it. You're going to break it open in that centrifuge tube. Then you're going to spin it again to isolate the cell gunk, the membranes, and all that stuff from the soluble stuff, and the soluble stuff should have your plasmid. And then you're going to transfer that to the column and do the washes to get rid of the other stuff, and then you'll elute or let your DNA out, and then go check the concentration and all that stuff. So you're going to need that intermediate tube that you've kind of roughly labeled so you can identify it from the other tubes and from other people's tubes, but it doesn't have to have all the specifics because you're going to toss it in just a minute. And so when you label though, you want to make sure that you're labeling the top and the side, especially when you're using these columns and these come in here, the caps can just pop off. So for each of your cultures, what you're going to need is you're going to need a column, you're going to need that tube that's roughly labeled, and then you're going to need a tube that is nicely labeled. But you can nicely label your tube during the weight steps when you're doing some spins, and you can even label the rough tube during the weight spin when you're doing your initial spin. But before you do any of that, you want to make sure you have all of the supplies that you need in order to do things, and that all the supplies are ready for you. And so in the kit, let's go through what's here and what you should have. So the first thing is you're going to be resuspending the pellet. And so you remove the media, you spin down, you remove that media, now you've got a pellet. That pellet is the bacteria that has the plasmid. Now we need to break open the bacteria and get the plasmid out. But we want to break them open in a solution that is nicer than the solution they're currently in, because we don't want to break them open until we kind of kill the stuff around them and protect them from breaking open too soon and get them in a nice pH and all that stuff. So this is resuspension buffer, this buffer P1. And often this buffer P1, you're actually going to find it in the fridge. And on top of here, there's a little check, and that check should be checked off. That check is saying, yes, I went ahead and I added RNase A. And since I added RNase A, which is an enzyme that chews up RNA, I need to keep it in the fridge because enzymes like to be cold. So it comes and comes at room temperature, but you have to add it, and then once you add it, you keep it in the fridge because that RNase is not going to be stable at room temperature in this buffer. And you want to put a check mark to indicate that you did add the RNase A. So now you might be asking, why do we add the RNase A? Well, we add the RNase A because we want DNA, and we want to get rid of the RNA so that we don't purify out RNA with our DNA and then confuse the DNA for RNA when we're doing stuff, or just have RNA that we don't want around. So the RNase A is going to help get rid of that, and then we will keep this in the fridge. So if you can't find your P1, don't panic, go look in the fridge. And so we'll talk more about what's in those liquids, but basically it's got some like glycerol and stuff so the cells don't break open due to osmotic stress. It's got a pH buffer, all that good stuff. So you're going to resuspend it in the P1, and then you're going to transfer it out of your culture tube and into an Eppendorf. That Eppendorf is where you're going to be doing the lysis. And this is your roughly labeled Eppendorf. So maybe you just have like a number or abbreviation, something like that. Something that you know and that is not the same as the person that's next to you doing the same thing at the same time. So you're like, uh-oh, what's a 1? If you have a lot of tubes, you can start by kind of vortexing them. But typically I just go, if you just have a couple, and just to take your pellet, you want to resuspend your pellet in the liquid. Just up and down until you've got it all resuspended. And then you kind of can just twist your pipette, the volume, so you draw it all out. And then take it and transfer it into that roughly labeled tube that you have. And so your rough labeled tube, now you can go and stick that in here. And you're ready to go ahead and do your lysis step. And now you want to add the lysis buffer. So the lysis buffer is going to be with these Keogen kits. The lysis buffer is your P2. And the lysis buffer, this is an alkaline lysis, and so alkaline, basic, and your basic alkaline conditions, DNA strands denature, so they come apart. And also the lysis buffer is going to have detergent, and that detergent is going to be dodecim sulfate, and so it is going to basically allow the membranes to come apart of the bacteria, help lyse the bacteria, and kind of sneak in between the membranes, lyse open the bacteria. The alkaline is going to denature the DNA, and your denatured DNA, well, now you've got all the strands come apart. what I like to do is just have my tubes open and then just squirt 250 into each of these, snap them closed, and go up, back and forth like this. And because you don't want to have cross contamination between your tubes, so but you don't want to also waste tips, so just squirt them in from above without touching anything.
Gotcha.
Okay, so let's start inverting it. Don't worry, we're going to get our plasmid put back together, but we're not going to put back together that big bacterial genome. And so the key thing with the Miniprep is prep. It's really, really cool in that when you denature or take apart the strands of the DNA, for a plasmid, it can quickly just go back when you change the conditions to be ideal. You get them out of that basic condition and into a neutral condition when you add the neutralization buffer. But for that genomic DNA, it does not have time to like link back up and stuff. So it just is gonna clump up. And so when you add the neutralization buffer, a couple of things happen. One is that your DNA strands come together, but another is that it actually has potassium. And potassium, so it's like positively charged. Dodecyl sulfate has a negatively charged head, so it's got this lipidy tail, but a negatively charged head that can glob on to that potassium, and it actually precipitates. So SDS page, so sodium dodecyl sulfate, we use that for protein gel electrophoresis because sodium dodecyl sulfate is soluble, and it'll keep your protein soluble and linear throughout the gel. Potassium dodecyl sulfate, KDS, is a whole different story, which you might have found out the hard way if you're trying to run SDS page and your sample is in a high concentration of potassium. So with the potassium, it's going to precipitate out the SDS, and the SDS is going to be bound to proteins and stuff. And so you're going to get this whole big clump of the denatured genomic DNA as well as the proteins and stuff all in just kind of like this KDS clump. Sometimes people add like thymethylene, it's like lyse blue. That is going to turn blue under alkaline conditions, those conditions you want for the lysis, and that'll indicate that you've actually lysed the cells open, or at least that the pH is high. And then you want to make sure that the pH gets high back to where you want it, and so that's where the color change comes in. Because it's a pH indicator, once that pH gets to non-alkaline conditions, to those neutral conditions, then it's going to turn clear, and when it turns clear, basically your stuff is just going to look white because there's all that pill gunk. It's like before, you just want to add it, like squirt it right from above. Okay. Awesome. Okay, so now we want to snap those closed. And you can already see that there's kind of that precipitate starting to form.
Oh, I see.
And so that's like your egg whites precipitating with that acetic acid. And so when you mix it, you'll see there's like, now it's getting, that blue color goes away, so we've got this colorless, but you've got all this gunky stuff. And so our goal now is to spin this down so the gunky stuff is all on the side. It doesn't clump very nicely on the tube, so when you're putting the tubes in, you want to make sure that you put it so that they're kind of like all oriented the same way. And your pellet is probably going to be more like kind of a smear along the sides because you have your tube directly in your centrifuge with all the caps arranged nicely. Now you know exactly where your pellet should be. You can go in there with your pipette tip and just go directly down and take it out without touching that pellet gunk, because that pellet gunk is what we're trying to get rid of before we go onto our column. And make sure that you're cross-referencing to make sure you're sticking it into the column that goes to that tube that goes to the tube that it came from. You should have your pellet. This one actually pelleted pretty nicely, but you can see there's a little bit on the edge of it too. That's done. We're gonna we want to remove all of it. So typically it says like 800 microliters, but basically you just want to remove it all without getting any of the pellet. Angle down and just directly take off as much liquid as I can without actually touching the pellet. And I'm just going to transfer it directly onto the center of the membrane without touching the membrane itself. Voila, and now it's ready to spin again. There's going to be a series of washes, and these are going to involve both binding buffer as well as wash buffer, and that is going to help your DNA bind, the other stuff not bind, and then you're going to elute or take out your DNA now nice and pure. There's the binding buffer, and that's going to help your DNA bind to the column and the other stuff not bind to the column. We'll talk more about the different conditions in these different buffers and why it makes the DNA bind and other stuff not bind. It has to do with various salts and isopropanol. And then you are going to have a wash buffer, so this buffer PE. This is another one where there's a check on the top of the bottle that you want to make sure you check. And so in this case, check is telling you that ethanol was added. So ethanol is going to help, it's going to help precipitate the DNA, but the salts and stuff will stay soluble, so this will allow you to wash those salts off. It's really important though that you make sure that you get all of the ethanol out, because we don't want that with our DNA. And so when you do the wash, there's like an extra spin step, and you may be like, why am I respinning it? You're respinning it to make sure you get rid of all the ethanol. When you're doing your spins and getting rid of stuff, again, you want to pull straight out of here. Avoid touching the bottom of here to the column tip. Excel, you want to just take it and dump it and carefully put it back in. If you're feeling really uncomfortable with that, you can kind of pipette things out too.
Yeah, so it's a lot nicer with the vacuum manifold, because then you don't have to, you just do it all. You add it, you turn the vacuum, it goes through. You do the next thing, it goes through. You still want to spin it though with the vacuum one at the last, like to get the ethanol all out. And then you also are going to spin it when you elute it, because the liquid's going out into the waste all the time with your vacuum.
The bottom line is that your DNA should be stuck on here, other stuff is not stuck on here, and now it's time to elute or get your DNA off now that you've removed all that other stuff. So often these kits will come with some sort of elution buffer, and so it's typically going to be like TE, so tris EDTA, it's like buffer EB. I typically just do it in water. You want to like let it sit on the membrane a little, let the DNA dissolve, and then take it out.
So now you're going to stick water directly onto the center of the membrane, but again, you want to do it without touching the membrane. Okay, and you want to make sure that, so it looked like there was a little water on the side. You want to make sure it's like directly on there, on the center of the membrane. Right now we're letting it sit so that it'll resuspend. Okay. And if the water's not actually on the membrane, then it's not going to be able to resuspend. And so we'll let them sit for a minute, and then you just kind of have to, when you put it in, you want to arrange it so that when it spins, it's going to be spinning so that the caps are going to be with the direction of the thing. Should be right in there, and you can go just toss the blue things, the membranes, because our DNA shouldn't be on there, and then just snap them closed, and we'll go and drop them.
Okay. And so at that point, your DNA should be nice and pure, and you can go and check to see that it's nice and pure by going and checking with a UV spectrophotometer, like a nano drop, and that will tell you the concentration of your DNA as well as the purity of your DNA based on a 260 to 280 ratio. So DNA absorbs UV 260 light. It also absorbs some 280 light, but proteins absorb 280 light better, and so you can kind of take that ratio of 260 to 280 to figure out how much, whether you still have protein left over. And then you also want to look at 230, because that's going to tell you about things like whether you have salts left over.
Karina's first mini preps. She did great. Lots of DNA.
So let's go look at what is in each of these different solutions, as well as a couple more tips to help your mini prep be successful. To review just the whole process and why we're doing this, so we've done some sort of molecular cloning probably, where we have a plasmid that has the instructions for making a protein that we want, or some other feature that we want, and it has an antibiotic resistance gene that will allow us to grow the bacteria that contain that plasmid in the presence of the antibiotic, but will kill off all the other bacteria so that only bacteria that have our plasmid will be able to grow. We call this antibiotic selection, and so we do the transformation. We stick the plasmid into the cells, we've plated them, and so then only the ones, so the plate contains the antibiotic that corresponds to the plasmid, and so only the bacteria that actually have the plasmid can grow. We let them grow on the plate, but that's not going to be enough bacteria for us. So now we're going to take that colony from the plate and stick it in some media, so like LB media overnight, about 5 mils. Let them make even more copies of the bacteria, making copies of the plasmid, so we get lots and lots of plasmid. And then we're going to do this mini prep where we're going to break open those cells and isolate out the plasmid so we can do whatever we want with it. And whatever you want with it might vary. It might be sending it for sequencing to make sure it's actually the plasmid, the sequence you want. It might be doing a quick test with colony PCR or analytical digest. Maybe you trust it and now you're ready to go and use it, say, for some subcloning, but you want to make some changes to it, or you want to stick it in a different type of bacteria that can express protein for you. Because typically when we're doing these things, we're doing the mini preps out of the cells that are actually really good, really high fidelity in terms of their copying. And so they're going to be the cells that are different than the cells that you would use for expression. And so these cells are going to be typically like XL1 cells or DH5 alpha cells, cells that are good at making a lot of the plasmid, but not so good at making protein. So now you're ready to do your mini prep. Got your starter culture. That was our amplification. We're going to resuspend it. So we're going to spin it down, remove the media, and resuspend our cells in the nice media that has the better conditions that we want to actually break them open into, so that when all those cell content, including your plasmid, comes out of the cells, it's coming out into an environment that is going to be happy for them and happy for you. And so it's not going to hurt your DNA and it's going to help you get the other stuff rid of. So now we're going to break open the cells now that we have it nicely resuspended in that buffer we like. We have sodium dodecyl sulfate, which is a detergent that is going to disrupt the membrane as well as the proteins. And then it's going to have NaOH. And this is where we get the alkaline part of alkaline lysis, is that we have NaOH, which is a base. And so it's going to raise that pH. High pH means low proton availability. We've got these conditions in which the DNA bases are actually going to not be able to bind to one another. So they're going to come up, strands of DNA come apart or denature. Then we're going to add potassium acetate. That's going to lower the pH to allow the strands to come back together. But that genomic DNA that you're trying to isolate your plasmid away from, that gDNA, it can't rezip in time. And so the potassium is going to precipitate. The potassium forms that KDS, potassium dodecyl sulfate, which is not soluble. That's going to precipitate the protein. It's going to precipitate the gDNA. And so all that's left in the solution is going to be your plasmid and all the other soluble stuff in the cells. At this point, you're going to go on to the purification. This is where you take the liquid that now has your plasmid DNA, so your pDNA, you're going to bind it to that silica column and wash it. So these columns, sometimes they have a cap on the top and sometimes they don't. But they all have this kind of binding membrane. You use very similar columns when you're doing things like a PCR product purification or an RNA purification. They have this silica column that is going to bind to DNA or RNA reversibly. This is the little spout you want to make sure that you're not touching and that you're not touching with your waste liquid, because that's where your pure liquid's going to come out at the end, as well as where your waste liquid's going to come out. You're going to have a waste tube that comes with it. And so that's kind of just your circular tube. It doesn't have a cap. And then you're going to want a cap tube where you're actually going to elute your liquid into. So these are typically used with a centrifuge spin, but you can also use it on a vacuum manifold, which is great when you have a lot of samples. It can really speed things up. For each sample, you're going to need those two microcentrifuge tubes, one that's roughly labeled for the lysis and the post-lysis centrifugation. So you want to have enough to differentiate it from the others, but don't bother with putting the date and stuff. And then you want to have one that's nicely labeled for the final purified product. So you want on that, you're going to have your plasmid name, the date, and your initials. You want to label the cap and the side, and then later you're going to want to put the concentration once you figured out the concentration after the purification. And then you're also going to need the spin column with the waste collection base. Don't write on the sides of this tube down here because those are going to get in contact with the ethanol. So just make a little mark on the top of the tube to tell you what it is. Speaking of what things are, let's go through the different reagents. And so you can prepare your own. So I'll provide a link, and you can also clean and reuse the columns. But often people use this Keogen kit, and there are kits from other brands, and they're all pretty similar. You want to make sure that whatever kit you're using or whatever homemade stuff you're using, you have everything before you start. And if you're looking for like one of them, like, where's the P1? The P1 is going to be that resuspension buffer that has the RNase A And or at least you add the RNase A, and then once you add the RNase A, that enzyme, you want to make sure it's kept cold, so that's going to be in the fridge. So look for it there. Also look to make sure that there's a check mark on the buffer PE, as well as the check mark on the buffer P1 telling you you've got the RNase A. The check on the buffer PE, that's going to be telling you that ethanol is added. It comes without the ethanol in it, so you need to make sure that ethanol gets added because that ethanol is going to be really important for keeping your DNA precipitated, but allowing the salts to go through. But it's not shipped with the ethanol, so you add the ethanol, it'll tell you how much you need to add, and then once you add it, it's really important that you put the check mark on so people know that you added it. So let's go through these steps step by step in a little more detail. So here's that amplification step where you're going to take one of these colonies. You want to take one of the nice big fat colonies. Avoid one of the colonies that has like little colonies around it. We call those satellite colonies. Avoid the satellite colonies too. Those can kind of grow taking advantage of the bacteria with the antibiotic resistance. They can, especially for some things like ampicillin, where the antibiotic resistance product is secreted, that's going to help things allow things next to it to grow. And so you want to avoid those and go for one of the nice big fat ones. Often you want to try to find one that's kind of isolated from other ones so you can pick just it, take your pipette tip, stick it in there, and drop it in your LB broth that contains your antibiotic. And then culture it overnight. Now you take your culture that grew overnight and you're going to spin it down. So you can spin directly in those culture tubes, even with the pipette tips in them. But just remember that when you remove the media, that the pipette tip's there, and so you typically have some sort of beaker or something with bleach in it that then you dump your waste into. But then you want to make sure that you don't just, when you dump that bleach into the sink, that bleach media into the sink, you want to make sure that you then get the pipette tips out for your own sake and for the sake of the other people working in the lab. So now you have done that and you've got this pellet, and our goal is to get it into a state where we can stick it on this column, but we're not ready to stick it on the column yet. We need to first resuspend it, lyse it, neutralize it, and then we can stick it on the column. So let's go ahead and get to that resuspension step. So again, we have our pellet and we want to resuspend it in the liquid that we like. So what is in this liquid that we like? Let's go through these. The first is going to be tris. So tris is a buffer, a pH buffer. It's going to keep the pH around 8. So a buffer is something that is going to have fairly even amounts of a conjugate acid and its conjugate base. So in this case, tris conjugate acid and tris conjugate base. And those are going to give and take protons to keep the pH steady where you want them. And depending on how much of the acid versus the base you have, you'll dictate what pH you have. And so here the pH is going to be about 8. And that's going to keep things happy for now. But then we're going to change it to those alkaline conditions, those higher pHs. There's also glycerol. So glycerol is going to help us keep the osmotic pressure intact so that basically osmosis, water will flow from a region where there's more water but less solute to a region where there's less water but more solute. And so as a result, what would happen if you didn't have that would be if you just had liquid outside, water would flow into the cells and burst the cells, and we're not ready to burst the cells yet. We want to first make sure that there's RNAs there and some other things there. And so RNase A is going to degrade the RNA when we open the cells, but it won't degrade DNA because basically what it does is it helps RNA attack itself with using RNA's OH group that DNA doesn't have. There's also thymolphthalate, which is going to serve as the pH indicator. It'll turn blue at high pH and then go back to colorless when we neutralize it. And be careful with the word colorless. So colorless is not the same as clear. And so I used to always get really confused, but clear is like transparent. Colorless just means like so white would be colorless. Now we're ready to actually go and do the lysis. So if we think about what we're trying to break open, in a bacterial cell, you basically have this bilayer membrane, and the inside of that membrane, you've got these lipid tails, which are hydrophobic, and so it makes you make this sandwich. And then they have these hydrophilic heads, and these heads are actually charged. So these like negatively charged heads of these lipid groups are going to be facing the watery exterior and interior of the cell. And there is cations like magnesium and calcium are going to stabilize that membrane by neutralizing those negative charges because like charges repel. We want to destabilize the membrane. So inside of this buffer, there's going to be EDTA. EDTA is a chelator. It'll bind to and hide those metals. And so it'll take those metals, and so those metals can't be used to help shield these negative charges, and the membrane's going to destabilize. You also have SDS. So SDS is going to be sodium dodecyl sulfate. And that is going to have this lipid tail and then this polar head, similarly to what we saw with the phospholipids. And so it's actually going to actually be able to sneak in there and break up the membrane. It can also go and glob onto proteins and help denature the proteins in the membrane and inside. And then you have the part that is actually going to be the alkaline part. You've got the NaOH, sodium hydroxide, that's going to disrupt the base pairing so that the double-stranded DNA splits up. So DNA is kept double-stranded thanks to hydrogen bonding between the bases. That hydrogen bonding, these non-covalent bonds, these attractions between these different bases are going to depend on their protonation state. Think protons, think pH. So at a low pH, where we were before, there's lots of free protons around. So these ends can get protonated and allow you to do that H bonding. But at high pH, what's going to happen is those are going to deprotonate, and then you're not going to be able to make those hydrogen bonds. So the strands come apart or denature. Note that you typically don't want to have a high pH with RNA because it can cleave, but DNA doesn't have this problem, just like they didn't have that problem with the RNase A. So we've unzipped the DNA and now it's time to zip it back up. And so we need to neutralize things. So remember we had sodium dodecyl sulfate before, which is soluble, which is why we use it for protein gel electrophoresis, because it's got that nice nonpolar part that globs onto those parts of the protein that are typically in the inside. And it's got that hydrophilic head. So what's going to happen is the protein's going to unfold or denature, but it's going to stay soluble thanks to that hydrophilic head. But the hydrophilic head is negatively charged, and so that would repel one another. And that keeps the protein linear to slither through your gel. Sodium dodecyl sulfate is soluble. Potassium dodecyl sulfate is not soluble. In this neutralization buffer, you have enough of the potassium acetate to provide enough of this potassium ions that are going to precipitate the dodecyl sulfate. And so you have this potassium acetate. The potassium part is what we need to precipitate things, and the acetate part is or the acetic acid part is what we need in order to get that pH back down lower to allow the bases to pair back up. So to allow that protonation, that lower pH, you have more of those protons, you get the protons, you get the hydrogen bonds between the strands. The plasmid can go and it can make all those hydrogen bonds, but that genomic DNA, that gDNA, you've got too much of a mess. It's too big, you can't zip up in time. And so instead, what's going to happen is it's going to get dragged down by that potassium dodecyl sulfate, which is not soluble. And that potassium dodecyl sulfate is going to be precipitating the membranes, it's going to be precipitating the proteins, and now it's precipitating that gDNA. And you just have this big old clump. The guanidium chloride is another salt that's in there, and that's going to help your DNA bind to the column. So before you can get your DNA onto that column, though, you are going to spin it so that you remove that gunk, and now you can move to the column. And this is where you need to be careful because the pellet might be messy and on sides of the tube. So you want to orient your tubes the same way. So I like to do it so the cap notches are on the outside, so you know exactly where to expect things. Then work quickly to outrace the pellet redissolving, and avoid touching that pellet with your pipette tip when you're transferring the liquid. But now you can go ahead and transfer it into your spin column. Then you take it for a spin, or you stick it in the column and you put it through the vacuum apparatus to pull the liquid through. If you're working with a vacuum, it's nice because the waste just kind of goes in the screen thing. But if you're working in one of these spin tubes, then you need to be careful about the waste is just going to go out into this capless waste tube. So at this point, you don't want it in one of your nice label tubes. You want it just in that waste tube that comes with the column. But when you pull the column out to get rid of the waste, you want to be careful that you're not touching this bottom part of the column to the waste. And that when you pour out the waste, you do it without kind of then putting the column back in and touching that. Okay, so now let's get the DNA binding. And so this is going to be very similar to with the PCR purification kits. And so with the PCR purification kits, though, there's often going to be a pH indicator that is going to be important for making sure that the pH is low enough for the DNA to bind. But typically we don't really worry about that in terms of these mini prep kits. And so what we're going to do is we are going to first get the conditions right for the DNA binding. And so we have this buffer PB. It's got an acidic pH. So at that acidic pH, not only do we have the DNA's strands together, but we also have protons protonating the silica in the membrane. That's going to neutralize the membrane. And that's going to make it so that the DNA can bind to it. So normally this would be negatively charged if you had a high pH. But at this low pH, the DNA is still going to be negatively charged. And so you want to make sure that they're not going to be repelling one another. Instead, you want the DNA to stick. And so by having this low pH, you're able to do that. We've got that guanidium salt, and what we call it is it's what we call a chiotrope. And so it kind of loosens up the water coat surrounding the DNA. And it's going to also denature the proteins. And DNA has that negative charge, and we shielded the silica by protonating it. These phosphate groups on the DNA are going to be way too acidic to actually be able to do that. So what we're going to do is we're just going to have the guanidium salt kind of shield it. And this is going to allow the DNA to get close to the membrane and to allow them to form more extensive bonding. To help them find one another, we've got isopropanol. Isopropanol is going to be less polar than water, so it's going to basically allow there to be fewer distractions between the DNA and the membrane, lower that dielectric constant. Now that the DNA is bound, it's time to wash it. And so now we get that PE. So you want to make sure that the check mark is on the top of the bottle, indicating that ethanol was added. The ethanol is going to dissolve the salts, but not the DNA. So our DNA is still going to stay stuck, but the salts are going to dissolve and then they're going to come off. Now, you want to make sure that the ethanol is all removed, and so that's why you spin it twice to remove the excess. So you spin it once after you wash it, and then you kind of like spin it again without having adding anything else, and it seems kind of weird, but that's just to remove all the ethanol. And now you're ready to remove your DNA. So what you need to do is, before we remember the high salt and low pH was helping it bind, so we need to get to low salt, high pH. At the high pH, we get the silica to deprotonate and become negatively charged. And if we remove the salt, well, now we've got this negatively charged DNA backbone and we've got this negatively charged membrane. They're going to repel one another and our DNA is going to come out. I like to do this in pure nuclease-free water. You just stick a little water on there. It'd be like 25 to 50 microliters, depending on what concentration you want and what you expect. Let it sit for a minute and then spin it to get your DNA out. And then it often also comes with like a buffer, such as like a TE buffer, where you can do that instead of water if you want. Then you want to go and check the concentration. So often this is done with a nanodrop. These are really cool, and what you do is it's a spectrophotometer, but unlike the typical cuvette-based ones, it makes a column that's vertical, and it does this with a tiny little drop so you don't have to waste too much of your sample. So you stick a drop of your solution here, you put down the arm, it pulls up really quick to make a little column, and then shines UV light through. The DNA is going to absorb UV light, and it absorbs best at 260 nanometer light, whereas protein is going to absorb better at 280, as well as 230. And so, but we kind of go by the 280 because it's more specific for the protein. 260 we look at when we're wanting to get the nucleic acid, and then we're also going to look at the 260 to 280 ratios, as well as 260 to 230. 260 to 280 is going to tell us about protein contamination. 260 to 230 is also going to tell us about salts and phenol and stuff. Phenol is less of an issue here, but when you're doing an RNA extraction, you might have phenol. But the salts are definitely going to be a problem that you want to make sure that is avoided. You can then go based on how much the DNA is going to absorb that light, and it's going to do so in a concentration dependent manner. So the more DNA you have, the more light that's going to be absorbed, and you can actually go and figure out what concentration the DNA actually is. And so typically these machines will just kind of spit it out for you these days. They'll also give you those ratios, and so for pure DNA, you want to look for a 260 to 280, about 1.8, and a 260 to 230, that's about above 1.8. You don't want to trust those ratios at low concentrations, and so don't worry about that. If your concentration is low, you have bigger problems with the low concentration, but don't worry about the ratios as much. If you got a low 260 to 280, you probably have some protein contamination. So maybe in the future, you want to be more careful when you're removing the liquids from your pellet. If you have a low 260 to 230, that could mean that you have salts left over from your extraction or purification, or carbs. So sometimes you do things like use a co-precipitant to help you precipitate a small amount of nucleic acids. Again, that's not typically something that you would worry about with a plasmid purification, but you might if you had another type of nucleic acid purification. And again, we measure at 260 for our nucleic acids because that's where we get the strongest, most specific signal. UV260 absorbance of 1 corresponds to 50 nanograms per microliter of double-stranded DNA, 33 nanograms per microliter of single-stranded DNA, and 30 nanograms per microliter of RNA. But typically the machines will just tell you to. And then you need to make sure that you tell future you, and so what you want to do is go and write that on the side of the tube in your lab notebook, and I like to keep it in a spreadsheet as well for easy reference later. So, hope that helps you appreciate the mini prep, and hope your mini preps are successful.